And the circuits in your cortex adapt instantaneously at the millisecond time scale to all of those demands. So when you walk from this building into the lovely sunny outside, or you go from reading a paper on the page to walking down the hallway, you don't need to grow a bunch of new neurons or new synapses to represent the dramatically different stream of inputs that your cortex is getting at that moment. And so there must be some mechanisms and circuit architectures sort of inherent to those circuits in the brain that allow them to perform such functional jobs, and also to be performing different computational operations essentially on a millisecond to millisecond basis. And the flip side of that, of course, is that we're also interested in what happens when that flexibility is lost, as we see in a lot of neurodevelopmental disorders. So in my lab, we are interested not only in the flexible circuit function that we see in the intact, healthy adult brain, but also the process by which those circuits develop to be that functional endpoint, in part because that gives us a lot of insight into what kinds of very precise wiring and local circuit architecture, what kinds of computational motifs do we see that are developing in the brain that will ultimately serve the flexible function of those circuits in their mature state. And of course, we always have an eye on the idea that disruptions of circuits, either in the mature state or during development, can very profoundly dysregulate the function of those circuits. So we see a dysfunction following either developmental perturbations or mature perturbations that could be either neurodevelopmental disorders, mutations associated with things like schizophrenia or autism, or also seizure disorders. And today I'm mostly going to focus on these top two pieces of the equation, but we're always interested in trying to learn something about function from studying dysfunction as well. So this is a highly schematized version of a local cortical circuit. And I'm highlighting here both the excitatory pyramidal neurons and three of the major flavors of GABAergic inhibitory interneurons. And one of the sort of most prominent theories in the field in the last decade or so has been that the diversity of these different sets of GABAergic interneurons is really the thing that allows cortical circuits to perform so flexibly. And the idea that these cells, because they have different intrinsic properties, different synaptic targeting, different firing profiles, they're engaged potentially at different times by different environmental cues or by different behavioral states, that they could essentially perform that flexible changing of circuit operations. And really the key idea that we try to drill down on here is the idea that the physical circuit in the cortex is always the same, right? You don't spontaneously grow a million new neurons every time you encounter a new environment. You actually don't even really grow new synapses that rapidly. But the functional circuit, the cells and synapses that are engaged and active at any given time point, that is flexible from moment to moment. And so what we want to do is understand better exactly how the functional circuit adapts on a moment-to-moment basis. Okay, so this is our favorite paradigm to use in the lab. This is mouse on a wheel. This is probably familiar to pretty much everyone here. And this has a couple of advantages. One of them is that mice very kindly will rotate between really quiescent states and very highly aroused states. And this gave us our first handle on how to identify circuits in the act of being flexible. So what we really needed was a paradigm where we could very carefully characterize state A and state B, and try to capture what changes in the circuit in the transition between those two states. And here, what we've done in a lot of our work that I'm going to show you at the beginning of this talk is just simply to use spontaneous behavioral state transitions as a way of capturing circuit flexibility. And of course, here, the mouse is quiescent, then becomes aroused and starts to run. And by now, it's been very well characterized that if you pay attention to this movie, you'll see some semi-externalized markers of behavioral arousal as well, because the pupil will be quite constricted when the animal sits still, dilates pretty profoundly when the animal is running. And so we can measure both locomotion and pupil diameter. Those give us two not entirely overlapping markers of the internal behavioral state of the animal. And of course, this paradigm is really adaptive. We can use this with in vivo electrophysiology. We can use it with imaging. And we also use it with behavior. And this data is just meant to highlight the fact that running and sitting does not fully capture all the things that are going on in the mouse's head. So on the x-axis here is normalized pupil diameter. On the y-axis is the amount of time spent in that state. And this is a lot of data from a lot of mice shown in distributions with blue and sitting and red is running. And you can see that although the pupil does indeed dilate quite a lot when the animal runs, there's a lot of overlap here in the middle, which suggests that internal arousal state and locomotion state are not entirely the same thing. In our early work in the lab, this is work that was done by Martin Bink and Renata Batista-Brito. We did a lot of electrophysiology in primary visual cortex using the paradigm I just showed you. So here pupil diameter is in blue, wheel position is in green. So when it's flat, he's sitting still. When it's wiggling, he's moving. Here's one example, single unit recording from V1 and local field potential recording on the bottom. Local field potential is, of course, really an aggregate measure of synchronous excitatory synaptic activity more than anything else. And what this really gives us is a bit of a snapshot into patterns of synaptic activity in the local network. What you can see is a very well characterized now response where the animal starts to run. Firing rates in V1 go up like a house. And the local field potential goes from having really big, slow, large amplitude events to very fast, high frequency events. And this is incredibly consistent every time the mouse runs. Okay. Unfortunately, this isn't going to have any audio with it. You'll just have to imagine that you're hearing the spike train. And this is sort of experiments we did trying to figure out if locomotion and arousal were really the same thing. And here we're imaging the pupil of the mouse as it sits on the wheel. The green line is wheel position. You'll see this never moves. The mouse never runs. The blue line here coming toward the middle of the screen, this is the timing of an air puff to the back of the mouse as he's sitting still. And we have local field potential and unit recordings down here on the bottom. And what you should be able to see here, although you probably can't hear it, is that at this point the animal's pupil dilates like crazy. The local field potential desynchronizes. We get lots of high frequency gamma activity. But in fact, actually the firing rates don't go up, they go down. So this whole series of experiments, and I'm not going to show you the entire data set here, suggests that actually running or locomotion and arousal have quite different effects on cortical activity. Nonetheless, they do both enhance the encoding of visual responses. This experiment is kind of interesting because it's a bit like sticking you in a dark room for a long period of time. You get really comfortable, and then every once in a while, at random time points, someone comes and breathes down the back of your neck. And what's amazing about this is that the mice don't run, actually. So if we look at the response to very simple drifting gratings, here we're looking at noise or background activity with respect to the amplitude of the signal or the response to a visual stimulus. This is just the raster plots from one individual V1 neuron. We can characterize the signal-to-noise ratio for different kinds of cells in the brain during different periods of time. So here A is aroused, pupil is dilated. Q is quiescent, the mouse has been sitting still for quite a while. Excitatory pyramidal cells are in blue. Inhibitory, probably parvalbumin positive fast spiking cells are in orange. And for all of our cell types that we've looked at so far, which actually extends beyond this data set, for both excitatory and inhibitory cells, the signal to noise of visual responses is always higher for animals when they're in an aroused state than when they're in a quiescent state. But that difference is actually considerably more profound for the excitatory cells than the inhibitory cells. And for those in the audience who think about things like population-wide correlation structure, we've also looked at the structure of noise correlations, which are slow time-scale correlated fluctuations between pairs of cells, and these are generally thought to be detrimental to neural encoding. And here, just for the excitatory cells, we find that noise correlations are lower in the aroused state than in the quiescent state, suggesting that arousal enhances neural encoding in at least two ways, an increase in signal to noise and a decrease in these background slow timescale noise correlations. Now, extracellular recordings like that give us a lot of information about the sort of overall population dynamics in different behavioral states, but they don't give us a very clear picture of exactly what's happening to the pattern of activity, and most importantly, the pattern of synaptic activity across the layers of cortex. And we know that there is a lot of information flow across layers of cortex in the cortical column. And to get a better snapshot of that kind of information flow, we've done a series of experiments. And this is a paired series of laminar probe and intracellular recordings. So we're putting in a patch pipette, recording one pyramidal neuron right next to a laminar probe, which is going vertically through all the layers of cortex. And then in this particular case, we're calculating a current source density profile, second derivative of the local field potential profile, which gives us something that is essentially a snapshot of the current flow across layers of cortex, which largely corresponds to synaptic current flow. And we can do this at the same time as our patch recording, and then what I'm showing you here is a spike-triggered average of the current source density profile across the layers. So this is layer 4 in the middle. The white triangle here is the position of the cell body of that layer 2-3 pyramidal cell from the example here, and time is on the x-axis. And so the dotted line here in the middle just represents the spike time of that excitatory neuron. This particular snapshot is not particularly illuminating because what it shows is a current sink right around the cell body of that neuron, so no surprise, cell is spiking when there's a lot of current flow around its cell body, it gets a lot of synaptic input. Okay. Things get a lot more interesting if we look at snapshots like this across different behavioral states where the animal is transitioning between a very well-characterized quiescent sitting state and a running state, for instance. And so these are two cells I'm showing you, each of them split into sitting and running snapshots. They're both layer 2-3 pyramidal cells. And again, these are spike-triggered averages, so the dotted line in the middle is the timing of the action potentials of the individual neurons. We can also do this for correlations with membrane potential activity, but I think these are a little more interesting to look at. So there's two things that are of interest here, right? So one is the basic pattern of synaptic correlated current flow across the lamina is quite different for different layer 2-3 cells. So these cells aren't very far apart from each other. They're both sitting probably in layer 3. But the patterns of activity that correlate with their spiking are quite distinct. The second thing is that in both cases, you can see a pretty profound shift in the pattern of laminar activity correlated with the spiking of that neuron as the animal goes from sitting to running, suggesting that it isn't just the average population responses changing with behavioral state. It's also the pattern of synaptic activity correlated to the activity of individual neurons across the layers of cortex. Okay, so sorry, this is cut off a bit at the bottom, but this is sort of a schematized version of our current model for how all these layers come together. We have arousal. You can measure that by either pupil diameter or wheel speed in this case. If the animal is running, we see a big increase in firing rate. If the animal is aroused in the absence of running, we actually see a bit of a decrease and a sparsening of activity in V1, especially in the superficial layers. But in both cases, we always see a signal to noise and an increase in the fidelity of visual encoding. Okay. So, not to belabor this, but we know that the cortical circuit function here is in fact highly flexible. It varies with behavioral state. The ability of neurons to encode a visual stimulus changes substantially depending on the behavioral state of the animal. We know that locomotion and arousal do different things to cortical circuit function. And of course, visual responses are enhanced when the animal is aroused. I think that makes sense actually to anyone who owns a brain, because if you sit in a large lecture hall for too long, you may find yourself zoning out. I think everyone has experienced periods of time where they were in a low arousal state and not encoding information all that well as compared to a highly aroused or attentive state. And so I think these are fundamental aspects of cortical circuit function, but they're quite well characterized at this point. Now, as I said at the beginning, my lab is interested in understanding a bit better the difference between the physical circuit and the functional circuit. And to do that, since we know that these circuits are flexible, we really need to capture maybe example populations of cells that could serve that function, right? If we're right that the thing that's happening when the circuit function and encoding changes with behavioral state, if we're right that the thing is that the functional circuit is changing, we should be able to find some populations of neurons which are active only at specific time points correlated with this flexible function of circuits. And so we started by looking at the VIP cells, which I know many people here are also interested in. VIP interneurons are extremely sparse. They're 1% to 2% of all the cortical neurons in total. Nevertheless, I'm going to show you some data suggesting that they pack a really immense wallop in the circuit. They're interesting to us in part because they represent this really interesting integration node in the circuit. They receive really substantial neuromodulatory input, both cholinergic and serotonergic, as well as local and long-range glutamatergic inputs, both top-down feedback inputs, local corticocortical inputs, and direct thalamic inputs. And then they make these really interesting and possibly opposing projections within the local circuit. They directly inhibit the apical dendrites of pyramidal neurons, and they also strongly inhibit intermediary interneurons, the somatostatin cells, which themselves innervate the dendrites of the pyramidal cells. We thought that since these guys get so much diverse input, including neuromodulatory inputs, they're a good potential candidate as a population we might be able to capture in the act of being flexible. And so this is GCamp imaging in VIP interneurons in an awake-behaving animal. On the axis here is speed of the animal on the wheel. So if it's flat, the animal is sitting still. And hopefully what you can see in the video is that in anticipation of movement, time-locked to the beginning of arousal, actually, the population of VIP interneurons comes on like a house. They're very, very active for a period of time, and then they essentially fade into the background and become very quiet, not very active when the animal is quiescent. And so this is a great example of a neural population, inhibitory interneurons, with strong synaptic connectivity in the local circuit that are, in fact, only on at key behavioral state transition moments. All right, so I mentioned at the beginning that we were also interested in how circuits develop to have these flexible components. And the data that I'm going to talk about in this next set of slides is a set of data where we used a targeted mutation early in postnatal life to disrupt the activity of the VIP interneurons in the hopes of unpacking what their function is, both during postnatal maturation of cortical circuits as well as in the adult animal. So ERB4 is a really important developmental gene specific to inhibitory interneurons. It's been well characterized in parvalbumin interneurons, has the same properties in VIP interneurons. I'm not going to go into all the details today. But it controls the formation of excitatory synapses onto the interneurons and the formation of inhibitory synapses from the interneurons to their targets. So mutating this gene in a Cre-dependent manner in the VIP cells restricts the driving excitatory input they receive and also their impact on the local circuit. So it's a bit like turning down the gain on these cells in the early postnatal period. So we made a series of mice with this mutation in the VIP cells, and then we looked at activity of pyramidal cells and interneurons in the cortex using both electrophysiology and two-photon imaging. Okay. We looked at a variety of different time points, so I'm highlighting 3 here. This is with electrophysiology, mostly recording pyramidal neurons in the cortex, starting at about P15. So 2 weeks after birth, the animals look completely normal. No different from the controls. And here, mutants are always in blue, controls are always in black. Firing rates on the y-axis. By about the adolescent period, about P25, we found a significant elevation in firing rates of the pyramidal neurons. That trajectory continued on into adulthood. By the time these animals are adults, there's a 6-fold increase in firing rates of excitatory neurons in visual cortex. That disruption extended to visual stimulus responses. So here we're showing just a very simple drifting grading to awake behaving animals on that wheel that I showed you earlier. This is a normalized histogram of visual responses of pyramidal cells, so normalized to background activity in the dotted line here. Same on the bottom for mutants, and you can see there are sort of residual visual responses here, but overall the responsiveness of the pyramidal neurons is pretty compromised. And this is an example of population histogram from adult mice. When we again looked at P15, so the end of the second postnatal week, we found no difference between controls and mutants, so that animals are pretty normal. Again, by mid-adolescence, what we find is that there is a halving of the selectivity of neurons in V1 for visual stimuli, and that continues on into adulthood where visual responses are still constrained. Now, there's always an issue with doing conditional line crosses like this. You may mess up the brain in all kinds of ways that you don't intend. And to control for all of those possible effects down in the brainstem or the amygdala, what we did was to rescue some of our mutants by reintroducing a functional ERB4 gene only into VIP interneurons in the cortex with a virus. We did that at about P5. So by the time we got to about P15, our replacement gene for ERB4 was expressed at normal levels, specifically in VIP cells in the cortex. That viral rescue completely rescued the firing rates of excitatory neurons in the mutants starting in adolescence and extending into adulthood. So the phenotypes that I just showed you are in fact the result of messing up VIP cells in the cortex. And one of the interesting things about this data is that if you look a few hundred microns away in these mutants, you find the cortex is completely messed up. The pyramidal cells a couple hundred microns away have incredibly high firing rates, and everything lacks visual responses. So this is a very local rescue, actually. Nevertheless, for the area where VIP cells are rescued, that rescue virus also replaces visual responses to drifting gratings back to control levels. In the adults, we find equivalent selectivity for visual stimuli in rescued mutants and controls. Okay, so this is no fun if you can't connect the activity of cells in the cortex to the animal's ability to perceive stimuli. So we took a series of these mice and we did our very standard, very simple go, no-go paradigm with them. In this task, the animals are trained to lick for water, and then they see either a go stimulus, which is a drifting grating on the screen, or they see a no-go stimulus, which is a blank screen. The go stimulus varies in contrast from below the perceptual threshold to about 100% contrast, so they get easy trials and hard trials. If they lick in response to a no-go stimulus, they just get a timeout. If they lick correctly in response to a visual stimulus being presented, they get some water, and these guys are water deprived. So this is a mouse going through one trial of this task. So he's head posted on the wheel. He's waiting for visual stimulus to come up on the screen. Visual stimulus shows up right about there. He licks it, gets some water, and then settles down to do his next trial. Mice will do about 300 trials of this before they get bored. So they do about 300 trials a day. Now, I'm not going to show all the data that we have on this kind of task. It turns out that simple detection of a high contrast moving stimulus is not V1 dependent, so animals can do the very simple version of this task with no cortex at all. However, the version that we're using here, which has both low contrast and high contrast trials, does require visual cortex. And in fact, what you see, and these are control animals on the left, so contrast is on the x-axis, and hit rate or performance is on the y-axis. And here I've divided the control data up into early and late sessions, so early naive animals and late expert animals. You can see that this is the psychophysical curve of the animal. Looks pretty good even in the early sessions, but it shifts to the left pretty significantly as the animal becomes an expert. So there's a perceptual learning aspect to this task. That leftward shift is entirely V1 dependent. If you remove V1, if you lesion V1, if you interrupt V1 function, even in expert animals, you lose this leftward shift, which represents a decrease in the threshold for perception. Okay, now, in contrast, our mutant animals, which have perturbed VIP interneuron function, do actually perform this task even in the early stages. They have a decent psychophysical curve, but they never show the V1-dependent leftward shift or decrease in the perceptual threshold, suggesting that the compromised function of VIP interneurons in the cortex has also compromised the animal's ability to perceive and respond to visual stimuli. Okay. Yep. It depends on your definition of learning, right? So they can learn the task, right? They give us a pretty good psychophysical curve. And that psychophysical curve is there, the 1 to 2 days. And this is after several other shaping series. So they've learned to respond to a drifting grading prior to doing the full task. So they can learn to do the operational part of the task. They obviously are responding to the visual stimulus. But that seems to be distinct from the perceptual learning process, right, which is that leftward shift in the visual response in the psychophysical curve on the previous slide, right? And I think those represent two different learning stages, in fact, and we can differentiate them based on their requirement for cortex versus the colliculus and other areas of the brain. Okay, so going back to our circuit diagram here, we've disrupted the VIP interneurons. But they have this extremely complicated set of inputs. And we really don't know whether the changes in the circuit that we're seeing are because the VIP cells are not responding to state change or to visual inputs appropriately, or if it's something downstream from the VIP cells. And to try to unpack that a little bit, and I'm not going to spend too much time talking about slice physiology, but to unpack that just a little bit, we did some slice physiology in our controls and mutants, recording from the VIP interneurons themselves, and these are just mini experiments where we're recording spike-independent mini EPSC events, and so this just characterizes some estimate of the level of glutamatergic input to these neurons. And we see a decrease in amplitude and an increase in interval for mini EPSCs into the VIP cells, suggesting that in fact they are receiving less glutamatergic input than they would normally. So excitatory drive to the VIP cells has dropped. However, we know they get strong cholinergic input. That does not seem to be affected by mutating our target gene in the VIP cells. Cholinergic response amplitudes were completely normal in the mutants as compared to the control. So this seems to be a selective loss of glutamatergic drive to the VIP cells. Now, in our normal animals, we find that the activity of the population of VIP cells is highly tied to the onset of arousal or locomotion. And here I'm showing you cells on the y-axis and time on the x-axis, and the dotted line is locomotion onset. You can see that most of our population of VIP cells turns on as the animal starts to move. In our mutants, we find that the state dependence of VIP cells has completely been ameliorated. There are some cells that still respond. Overall response amplitudes are quite low, and many of the VIP cells don't respond at all as the animal becomes aroused or starts to run. So these cells no longer respond to key behavioral state transitions. So the question, of course, is, okay, if the VIP cells are not doing their job and they're not active at key behavioral state transition points, what's happening to the pyramidal neurons? So we went back to doing electrophysiology for this part of the experiment. So these are extracellular recordings of pyramidal neurons throughout all the layers of cortex. An example on the left with the raster plot and histogram, a population average on the right. So this is average around the time point of locomotion onset, showing population increases in firing rate in anticipation of running, and some sustained firing during running. And that's controls on the top. If we record pyramidal cells in our VIP mutants on the bottom, we find that these mice have actually only one cortical state. We don't know quite what it is, but it's very, very stable. So the VIP mutants here show no cortical state transitions at all, no state-dependent modulation of the firing activity of pyramidal neurons. So measuring this by modulation of firing rate activity, we find that the modulation of pyramidal cells following VIP perturbation has dropped almost to zero. Okay. So the data set that I just showed you suggests that the VIP cells have been disrupted and their input is disrupted, their output is disrupted, the pyramidal cells have lost their state-dependent transitions. But VIP cells have really interesting projection patterns in the local circuit, right? They have these opposing direct inhibitory connections as well as this potential for disinhibiting pyramidal neurons. What we see in the pyramidal cells is really an increase in firing rate activity, and we thought that this might suggest that disruption of the VIP cells had released the somatostatin cells, and that might have caused over-inhibition of the pyramidal cells, but they didn't match with our firing rate data, and that made us go back and try to figure out exactly what had happened to these intermediary parts of the circuit following VIP interneuron disruption. So we went to look at the somatostatin cells. To do this, we had to change to an intersectional genetic approach. In this case, we're using a cross between a Cre line, which is carrying our mutation, Cre-dependent mutation of the VIP cells, crossed to a somatostatin flip line, which allows us to mark these somatostatin cells with a flip-dependent YFP so that we can record from them both in slices, and I'll show you in a moment for some in vivo imaging. Again, we're looking at spike-independent release of neurotransmitter here, but in this case, we're looking at GABAergic transmission. And we find that the somatostatin cells show an increase in interval between mini iPSC events corresponding to a decreasing GABAergic input. So this suggests that the somatostatin cells indeed have lost a lot of inhibition coming from the VIP interneurons in our mutants. Okay. Now we can take the same intersectional genetic approach. So here we are using a flip-dependent GCAMP6 made for us by Carl Deisseroth's lab. In normal somatostatin interneurons in V1, the entire population is highly visually responsive. So these bars on the bottom just represent the timing of visual stimulus presentations on the screen in front of a mouse on the wheel. Looking at the somatostatin cells in layer 2-3, in our control animals, this is actually a pretty small data set, we have a lot more data now. In our control animals, we find that on average, somatostatin cells are not very modulated by behavioral state transitions at locomotion onset. They show a small positive bias towards increases in firing rate as the animal starts to run. In comparison, in animals in which the VIP cells have been dysregulated, we see incredibly enhanced somatostatin interneuron activity following the onset of locomotion, suggesting that loss of VIP inhibition allows the somatostatin cells to kind of break free and be more active than they should be right at these moments of arousal and activity. One of the other things that happens to the somatostatin cells in the absence of appropriate VIP cell inhibition in the network is that their very strong, very powerful population-wide visual responses basically go away. So in control animals on the left, you can see responses to drifting gratings. These are just example cell traces, calcium transients from individual somatostatin cells. On the right is a similar number of cells, but from an animal in which the VIP cells have been dysregulated in development. I'm showing pretty good locomotion responses here. The locomotion periods are in gray, but no visual responses left at all. Here's signal to noise for the visual responses in the somatostatin cells. In controls, is pretty robust. In the mutants, has gone almost to zero. Okay. So kind of to put all those things together, right? The VIP cells represent this incredibly tiny population of cortical neurons, and yet they appear to have an incredibly powerful role in the way that the circuit develops in postnatal life. So disrupting these cells appears to dysregulate pyramidal neuron activity profoundly and also causes a loss of visual responses and appropriate state-dependent responses in the somatostatin cells. But, right, so the data that I just showed you is also very unsatisfying in another way, and that is the use of these conditional line crosses, right? So it means that we are causing a disruption of some particular cell type. It only happens at one particular developmental time point, which in this case is the end of the first postnatal week, and we really have no control over when our manipulation of cortical activity is happening. It doesn't allow us to pinpoint the developmental window in which those VIP interneurons might be critical. So to kind of get around that constraint, we switched approaches and did a series of experiments where we injected V1 of VIP Cre mice with a Cre-dependent KIR 2.1, which is a potassium channel. So we're overexpressing a potassium channel in these cells, causing them to hyperpolarize and fire less than they should. And we're doing this at 3 different developmental time points. So we've done this at P2, P10, and P50. And then we've let the animals grow up to maturity between P80 and P100, and recorded activity in V1. Now, one of the things to remember about looking at data like this is that it takes approximately 7 days for the potassium channel to reach peak expression. So you have to add about 7 days to each of these time points to know when the perturbation that we're inducing really takes place. Nevertheless, we get really nice expression. So this is an animal injected about P2. You can see VIP cells in red, KIR 2.1, GFP in green, and nice overlap, and this was after the animal grew up to adulthood, so this is a long-lasting expression. Okay. So here we find examples from a P2 population of injected mice injected at P2, right, with KIR 2.1 just in the VIP cells. I'm showing you distributions of interspike intervals. And you can see that when the animal gets to be adults, the pyramidal neurons show a very different distribution of firing rate activity than they would normally. And in fact, firing rates in these animals are upregulated by about 4 times. So this nicely recaptures some of the effects of our targeted mutations from that last data set that I showed you. However, when we do this manipulation at older time points, we find no effect. So this role of VIP interneurons in regulating the firing of pyramidal cells appears to be restricted to a very specific postnatal time point. We can do the same thing looking at state-dependent regulation of cortical activity. And here again, I'm showing you example histograms of pyramidal neuron recordings from animals that were injected with the KIR 2.1 virus at P2. So these are our control animals up here on top, showing the sort of now traditional increase in firing rate with locomotion onset. In comparison, our injected animals down here on the bottom show no state dependence in that chunk of V1 cortex at all. So measuring this as a modulation of firing rate activity, we find robust modulation in the controls, almost no modulation at all in animals injected at P2. And again, we find no impact of doing this manipulation in older animals. So this is, again, restricted to an early postnatal time point. And our third way of assaying the function of the system is obviously to present visual stimuli. So in our control animals, we get a robust visual response to drifting grading stimuli. In our KR 2.1 injected mice, early on, we get no visual responses at all from pyramidal neurons in these mice after they grow up, suggesting a really significant decrease in visual responsiveness, and no impact on the visual response of pyramidal neurons in adult animals. Okay. Now, there's a caveat to these data. There's always a caveat. No matter what you do, there's always a caveat. But the caveat to this particular data set, right, is that I'm aggregating a lot of data here, so I'm showing you a lot of average responses. And in a minute or two, I'm going to unpack this a little bit more, because this is actually a fairly surprising finding for us. Right, so this suggests two things. We have a really consistent early postnatal window for a purely developmental role of VIP interneurons in setting up this functionally flexible circuit for adulthood. But two, we're not finding any impact of dysregulating the VIP cells on average in the adults. And that's a bit surprising because we think that they actually do have really profound roles in the circuit in these sort of state-dependent transitions in adult animals as well. So I'll come back to that in just a moment. Mm-hmm. Yeah. The numbers suggest no. The very boring histology slides that I am sparing you at the moment suggest an equivalent number and density of expression across cells. And the cellular level impact appears to be the same if we patch cells from early or late injected animals. They appear to be responding the same. Because the KR expression doesn't really peak for about a week, the impact of that expression is really after the time point when any shift in the role of GABA release in the cortex would have happened. So really the peak impact of the KR 2.1, even at those earliest injection time points, doesn't happen until about P10 or P12. So it's a pretty consistent impact at the cellular level all the way up through adulthood. Okay. All right, so data like this sort of suggests that there is a very specific critical window for VIP interneuron roles during this postnatal development, right around the time of eye opening, in fact, from about P10 or P12 up to maybe P18 or P20 at the latest, where the 1 to 2% of cells that are the VIP positive interneurons really control the development of functional cortical circuits. Later on, dysregulating these cells does not appear on average to dramatically dysregulate the rest of the circuit. But in this critical window, which is an interesting time point, in fact, because we know that somatostatin interneurons are incredibly important in the first postnatal week for setting up thalamocortical wiring in the cortex and for several other wiring events of appropriate connectivity. We know that PV-positive interneurons are important from somewhere in here to about there for critical period plasticity. The VIP interneurons appear to be sort of in an partially overlapping period, kind of right in between those two time points. But we know that they're important for development of visual responses, overall firing rate dynamics, and state dependence. Okay. So coming back to the idea of adult circuits, we were really perplexed by the lack of average impact that we found in our adult animal manipulations. And we went back to try to dig into that a little further. And we switched to using a slightly different manipulation here to take advantage of both intersectional genetics and GCAMP imaging, as well as targeted manipulations of a specific population of neurons. So you can ignore this. It's sort of like alphabet soup. In this particular case, we're using a VIP line crossed to the SOM flip line, and we're killing the VIP cell population locally with a Cre-dependent caspase virus. Query-dependent caspase takes something on the order of a week to 14 days to be fully effective. And once it is effective, it kills about 65 to 70% of the local VIP interneurons in the injection area. And then we can go and image the activity of somatostatin interneurons in that same area where the VIP cells have been eliminated. And so here on the left are control animals. So x-axis is time, y-axis is somatostatin cell number. And here again, we've aligned this to the time point of locomotion onset at the white dotted line. And in control animals, the somatostatin population, especially in layer 2-3, is just slightly positively inclined towards being activated at locomotion onset. But there are many somatostatin cells that are not normally activated by the onset of locomotion. In comparison, in our VIP caspase mice, what we find is that there are many more somatostatin cells active at locomotion onset than there would be normally, and overall activity in somatostatin cells has increased. This is not particularly surprising, given that we know that the strongest inhibitory input that the somatostatin cells get is the VIP interneurons. But overall, what we find is that the distribution of modulation in the control animals is centered just about here. In our animals where the VIP cells have been eliminated, the population shifts to the right. So on average, we see maybe twice as much modulation at locomotion onset in the somatostatin cells in animals where we've eliminated the local VIP interneurons. I think this is sort of interesting for two reasons. One, this is a bit distinct from some of the other things that I've talked about in that it's a very time-limited adult manipulation. Right? So it suggests that the VIP cells are, in fact, doing something very important in the adult circuit, which is a bit reassuring. But two, it really seems to be releasing the somatostatin cells at a very specific time point. So we don't see that the somatostatin cells are simply disinhibited all the time. What we see is that they have very specific periods where they're showing abnormal levels of activity, and those are time-locked to things like arousal and locomotion here. We know that the somatostatin cells also receive a lot of cholinergic inputs, which do depolarize them and can push them to fire. So one of the things that this might be doing is to unmask, in the absence of VIP inhibition, neuromodulatory enhancement of somatostatin cell activity around these important behavioral time points. Okay. Let's see what else I got here. All right, so just as a last teaser, we think that one of the best ways to unpack the rest of this story and to look a little more closely at the real-time interactions between different interneuron populations is to do it with time-limited and much more online manipulations. So this is a little bit of preliminary data. This is not really ready for prime time yet. Of a closed-loop system we've built for real-time manipulation of one set of interneurons, like the VIP cells, while imaging from a second set of interneurons. So our goal kind of going forward with this kind of project is to really pick apart the state-dependent interactions between different interneuron populations in the context of active behaviors or behavioral state, and try to better model and better understand how those two different pathways through the system, both the direct inhibition pathway from the VIP cells and that disinhibition pathway through the somatostatin cells, really play a role in modulating the activity of pyramidal cells online. Okay, and with that, hopefully that played. And with that, I'll just come back to the schematic that I put up at the beginning, which is just to sort of wrap up and say that what we're really interested in is the idea of flexible circuit function. It's not really a new idea, actually. It's one that our field stole from people who work on invertebrate nervous systems. But the idea that the physical circuit can be sculpted by things like neuromodulatory inputs or interactions between different cell types to produce a functionally different circuit that's adaptive to different streams of input from the environment, different behavioral contexts, I think is a really key element of cortical circuit function, one that is obviously heavily shaped by development, and we know is also shaped by dysfunction. But in this case, I think even unpacking the developmental interactions is complicated enough. So most of the work that we did was obviously not done by me. It was done by a series of extremely talented postdocs in the lab. The very early work was done by Martin Vink, who isn't even on this list anymore because he's been gone for a while. A lot of the VIP interneuron work was done by Renata Batista Brito, who's just left to start her own lab at Einstein. Most of the imaging work was done by Katie Ferguson, who's a very talented computational and experimental postdoc in my lab. Slice physiology was done in collaboration with Mike Higley's lab, and some of our reagents were made in collaboration with Carl Deisler's lab at Stanford. And thank you.
I don't know if I need it, but you mentioned that this the effect of the KR 2.1 silencing is developmental, but when you made this ERB4 animal, you could rescue the phenotype with a virus injection.
Early,
so
yes.
Yeah. So how do you have to inject the virus?
You have, yes, it's only rescuable if you replace the virus by about the end of the first postnatal week.
It was very interesting to see only one state in the pyramidal neurons in the case when you knocked out VIP neurons early. But at the same time, you had two states in the somatostatin cells.
Yes.
It was so it seems that there are still two states inside of the cortex, just that their effect on pyramidal neurons is somewhat different. Can you elaborate a bit on what are the circuit mechanisms which would allow those, not the absence of two states, but the shift of the states from one neuronal population to another?
So, right. So you want me to explain the brain?
Yes.
Okay. All right, so there's multiple layers to this, right? So one is that the intuition that we have from a lot of our experiments is that state in the cortex is a very local thing. So we can disrupt state-dependent transitions in this chunk of cortex and a couple hundred microns away in the next column over. Everything looks fine. So state obviously exists elsewhere in the brain, right? And there are state-dependent signals that are converging on specific target neuron populations in the local circuit to give you the state-dependent activity of pyramidal neurons. It's a bit hyperbolic of me to say there's no state in the brain in these mice, right? But if you just looked at the pyramidal cells, you would never be able to extract any information about behavioral state from them because they no longer have state-dependent changes or modulation in their activity. They also lose their locomotion-induced visual response gain. They lose all the other markers that we think of as being state-dependent regulation of activity. Now, there are a lot of neuromodulatory inputs to different subtypes of cells in the cortex, right? So the same cholinergic inputs that drive the VIP interneurons to fire also modulate the activity of somatostatin cells, while at the same time affecting the presynaptic side of the synaptic interactions between those cells and their targets, as well as the thalamocortical synapses that are providing visual inputs to the system. Right, so it seems likely to me that we can remove some layers of the state-dependent integration of signals, or the integration of state-dependent signals, while still seeing other layers of those state-dependent signals remaining. Now, when we use the pyramidal cells as the only readout for state, right, if we, let's say, increase the activity of somatostatin cells at these time points of arousal or locomotion, right, we're over-inhibiting the pyramidal cells, which would normally be showing an increase in firing rate. So it may simply be that in the absence of VIP inhibition, you have over-inhibition from the somatostatin cells, which is canceling out some other normally very important set of signals that's encoded by the pyramidal cells, either by over-inhibition of the dendrites, removing the dendrites as a computational compartment of the cell, or simply by canceling out other inputs to the cell. You know, those are questions that we really can't answer without going in with patch electrodes and getting a better grasp of what's going on at the membrane potential level in the excitatory neurons, which is something that we're doing, but it's a slow data collection process, right?
Hi. So in some of the earliest perturbations that you showed, there was this dramatic effect that the firing rate of pyramidal cells went up with age very significantly. So I'm curious, did you notice any epilepsy in those mice? And if not, how about any perturbation to the patterns of oscillatory activity? Any rhythms that were perturbed?
Those mice do not have seizures. They show the same degree of seizure resistance to chemoconvulsants as control mice, somewhat surprisingly for us. We see elevated firing rates in other parts of the brain as well, although nowhere as profoundly as the cortex. So we do see elevated hippocampal firing rates in those animals as well, but we don't, but not to the same degree as in a sensory cortex, although we do see like prefrontal cortex and sensory cortex are equivalently upregulated. So there isn't any overt pathology. I've forgotten the second part of your question. Oscillations, yeah. So oscillations are really interesting in those mice. There's a whole other story embedded here about gamma oscillations. So they do show normal levels of beta and gamma oscillations, but the entrainment of individual pyramidal neurons to those oscillations has been completely abolished. So they show no entrainment of local pyramidal cells to either gamma or any other frequency that we've tested. Honestly, they lose all their entrainment to low frequencies as well. That's specific to the pyramidal neurons. Fast-spiking parvalbumin and positive interneurons are still normally entrained for the most part. In fact, in those mice, PV interneurons appear to be unaffected and still show some evidence of state-dependent signals as well.
Hi, Jess. Is there any evidence whether the VIP neurons are signaling solely through GABA or also through VIP or some other peptide?
I love that question. We have absolutely no idea. And you're the second person recently to ask me that question. And you know the first person who asked me that question, right? So, so probably yes. Probably the peptide release is really important for the system. We don't actually know what peptide release during development versus adulthood looks like. We don't know how the firing of those cells correlates with peptide release. So for instance, it could be that the canonical view has always been that you get peptide co-release at very high firing rates, but we don't really know if that applies to every cell type. No knowledge whatsoever is something that we'd really like to take a look at. The only piece of evidence I have that the peptide is probably important is that the receptor for VIP, one of the receptors for VIP, has come up several times in studies of genetic risk factors for schizophrenia. So knocking out the receptors for VIP has a profound impact on the brain, but we don't really know anything about the dynamics of the peptide, or somatostatin for that matter. All right, thank you very much.